Hi guys, this is Module 2, Section 2. This lesson we are going over electricity in the brain. So last time we introduced the action potential. We'll go over some electric principles that can help us understand the action potential more deeply. Then we'll review electrochemical gradients and finally synaptic transmission. So we know that the action potential is the term given to the signal that neurons conduct to one another, and that the signal that is passed along is an exchange of charged ions across a membrane, and that signal is by nature electric. In this lesson, we aim to cover some general principles of electricity that can help us understand the action potential, what is meant by resting membrane potential, and how we can manipulate the electric properties of the neurons to study them. So first, for some quick definitions that most of you probably already know, but we want to get everyone on the same page. There are three main characteristics of electricity that are essential in understanding how the action potential is an electric signal. So for a moment, picture a group of molecules. Some are positively charged and some are negatively charged. We know that opposites attract, and so for any separation, the positive molecules will want to move towards the negative molecules, and vice versa. This attractive force, in the case of electricity, is called electrostatic force. We can think of these positive molecules as having kinetic energy in their movement towards the negatively charged molecules. Electrostatic force is conservative, so when a capacitor is introduced to the system that separates the charges, due to that separation, we have what's called a potential. So it's like potential energy. We can measure the amount of work that is being done to keep the molecules separated. So voltage then is a measure of this potential energy between 2 points. Formally, 1 volt is 1 joule per coulomb, which tells us that it's a measure of the amount of energy associated with one charged molecule. So when a molecule of one charge is moving towards one with an opposite charge, the kinetic energy the molecule now has is called current. In order for a current to move through an electrical system, there must be an uninterrupted pathway for the charges to flow. Current is typically measured in the direction of flow of the positive charges and follows the path of least resistance. The path of least resistance is also known as the path of greatest conductance and is a material's ability to conduct electricity. So just quickly putting current in more chemical terms, I suppose. Like I said before, the direction of current is defined as the direction of positive charge. So in terms of chemical molecules, these will be cations. Cations are positively charged molecules, and so of course they will move in the direction of electrical current. Anions, being negatively charged molecules, will move in the opposite direction. So when we talk about depolarization, this is a reduction of charge separation. And that's why it's changing that potential. It's changing that membrane potential. And that leads to a less negative membrane potential. And then hyperpolarization is an increase in charge separation, leading to a more negative membrane potential. And the cell membrane potential results from the separation of net positive and net negative charges on either side of the membrane. So let's take a look at what that looks like. From the previous lesson, we know that the membrane has a resting potential of negative 70 millivolts and that the action potential occurs when the membrane is depolarized to threshold, which for most neurons lies around negative 50 millivolts. We can apply the concepts of potential across the membrane, being a measure of the difference between charges. Instead of electrons, as in the case with electric circuits, the membrane potential is created by the existence of different concentrations of negatively and positively charged ions on either side of the axonema. The space inside the axonema is generally more negatively charged, and outside the axonema is surrounded by positively charged ions. It is this difference in ions that creates the membrane potential. The resting membrane potential is maintained through the movement of ions through ion channels, which we'll go into more depth about on this slide. So at resting state, like we said, the inside of the neuron is more negative relative to the outside. And this is due to highly negatively charged proteins or chlorine ions those sorts of negative charges. So although the concentrations of different ions attempt to balance out on both sides of the membrane, they cannot, because the cell membrane only allows for some ions to pass through channels. So at rest, potassium ions, which are found on the inside of the membrane, can actually cross the membrane easily. So they're going to be moving out of the axillemma pretty easily. And so we already have a high concentration of sodium outside the axillemma. So when there is a low concentration of when there's a low concentration of potassium inside of the neuron, this creates a negative environment relative to the outside. So just putting that into different words, passive movement of potassium out of the resting cell through ion channels balances the passive movement of sodium into the cell, because it's more difficult for sodium to move into the cell. This would technically eventually run down and the membrane potential would be gone, but we have something called the sodium-potassium pump, and it basically pushes ions in the direction that they don't want to go. And we'll talk about more what this means in a future lesson, but the sodium-potassium pump is largely responsible for maintaining the resting membrane potential. So this slide gives you a pretty good idea of what's actually happening as the membrane potential, as the action potential passes down the axillemma. So the membrane potential is modified through movement of ions through ion channels. When the neuron receives a signal, voltage-gated channels are opened, causing depolarization of the membrane. This in turn opens more channels and allows for further depolarization. And so in this way, the action potential can be thought of as an electrical impulse with flow of ions instead of electrons. So as you can see, as these channels are opening, we have positive charges rushing into the cell and negative charges rushing out of the cell. And then, of course, it returns to normal after. In a regular neuron, there is, a brief period where the cell is positive inside past when the action potential was there. That's the refractory period. But I think this graphic just gives you a good idea of how the action potential moves and what it looks like when those voltage-gated channels open. So why do we care about this, right? The electrical properties of the action potential can be harnessed to study the activity in a neuron. Tools that measure the voltage across a membrane allow us to know when a neuron is firing and when it's at rest. This also means that the neuron will respond to electrical stimulation, given that the channels that open upon action potential propagation are voltage-gated channels. By artificially manipulating the membrane voltage, we can cause or prevent action potentials. And this concept is used everywhere in neurotech, as many methods of recording neuronal activity involves picking up electrical signals. Yes, and finally we have our summary slide. So the resting membrane potential is created due to differences in the concentration of positively and negatively charged ions inside and outside the axillemma. In the action potential, these charges are briefly reversed, which opens voltage-gated channels, allowing for the propagation of the action potential down the neuron. At our next lesson, we will be going over the driving force behind the action potential, or electrochemical gradients.